We have William Valder present the talk, even jointly with Lisa Tarantino from University of North Carolina.
Okay. So I'd like to thank the organisers for inviting us here for this very interesting conference. Reproducibility and replicability is always in the back of our minds, but this has definitely brought it to the front of our minds for this talk. This talk will be partly conducted by me and partly conducted by Lisa Tarantino. Not sure how that's going to work, but we'll see. I'm going to start off with a few slides just on the motivation of the talk. Mouse behavioral models rely on the assumption that the phenotype is in some sense stable, at least in the sense that if you were really able to fix the surroundings, the protocol, the intervention that you applied, and also the mouse strain, then the phenotype values will be the same on average. And in behavioral mouse genetics, we're usually interested in knowing whether and how these mean values are affected by difference in genes or sets of genes or genomes. And in the simplest case, we would compare phenotypes from different strains using some sort of standard type of procedure such as ANOVA and look for whether the underlying strain means are different. And then we might go back and try to relate those to shared genetic features between those strains. All of the tests used, and certainly the ones that are most commonly used in behavioral genetics literature, assume that there is homogeneous variance, homoscedasticity. That is, that all of the strains have the same within-strain variance. This homoscedastic assumption, although it's obviously false to some extent, is tolerated in practice because I think it's assumed that within-strain variances are assumed to be only very slightly different. But actually strong variance heterogeneity exists. And it can arise through a number of sources. The most sort of mundane might be a choice of transformation, or indeed a choice not to transform. So we might think of a phenotype that we're measuring that has an underlying mean variance relationship. You increase the mean, you just naturally increase the variance, and if that's the case, then sort of readings, observations from higher variance from higher mean strains will just be more variable and sort of less precise. Also, of course, confounding with sort of animal handling and so on. Those sort of uncontrolled experimental covariates in a poorly designed experiment might confound with particular strains. And then there's direct effects of genetic background itself on the variability of a phenotype, and that's going to be the focus of this talk. What are the implications of having strains with different variances? Well, in studies that test for features that are common across many strains simultaneously, the fact of heterogeneous variances can mean that variance in some strains, particularly high variance strains, is going to be underestimated. Most of these models assume that there's just one variance, and so if you're not accounting for that, then there's going to be model misspecification, and this can lead to studies where significance tests are biased, in that they often they might be anti-conservative, and so findings may fail to replicate more often than you would think, because the original studies were kind of unstable in the sense that they relied on something where there was some high variance strains. And you can think of a high variance strain almost as like introducing outliers into a study. Knowing about strain variance, being sort of cognizant of, okay, this is a high variance strain, this is a low variance strain for a particular phenotype, may help you with decisions about how to design an experiment. For example, you could see that strain as being, if you're interested in mean effect, you could see that strain as being something where you have to phenotype more of them to get an equivalent level of precision on their strain mean. You might want to avoid those strains entirely, or you might be interested in strain variance in itself. So the plan of this talk is that Lisa is going to describe two studies, really sort of one's almost a complete replication of the other on the open field test. And then I'm going to talk about genetic effects in general on variance and do a bit of modeling of some of the strain data. Lisa's then going to hone in on one particularly high variance strain, which is C58J. I'm going to say a little bit. We have replication of numbering. That's unintentional. And then I'm going to say a bit about variance as a nuisance parameter and how it might guide our design. So I'm going to hand over to Lisa. Well, certainly no questions yet. I haven't really said anything.
Variance was an unbiased measurement. So what? I understand the standard deviation is a biased estimate, and we make corrections for low n. For example, with an n of 2, the correction is about 25%. But I don't understand why variance is biased.
No, it's not that variance is biased. It's that if you're using a model where you're assuming that where say you're comparing groups, such as an ANOVA model, there's only one residual variance in there. There's only one assumed residual variance in there. You're not assuming that the model doesn't accommodate the possibility of there being a different variance for group 1 and a different variance for group 2 and so on. So they're assumed to have the same variance. That's one of the kind of
okay,
assumptions.
that's the point we will return to. It's not a trivial one.
Okay, so obviously Will and I are from two different worlds, but we managed to communicate and talk about the kind of mix of behavior and statistics and how to best design experiments and also analyze data. My laboratory has the ultimate goal of identifying genes that influence behavior. In particular, we're interested in anxiety and depression, stress, and also addiction. And we use a number of forward genetic approaches, including standard quantitative trait locus or QTL studies. We've used ENU mutagenesis and also doing inbred strain surveys, initially with the hope of performing genome-wide association studies, but also as a way to capture the wide range of phenotypic variation across a number of different genetic backgrounds and to get more of an idea of what exists among these inbred strains for more further analysis. So that's what I'm going to talk about today. We had conducted 2 different inbred strain surveys of open field behavior. And the first study, which was actually conducted in my laboratory but was a grant funded to Tim Wiltshire, was done at the Novartis Research Institute or Foundation in California. So that I'll sometimes refer to as the GNF study, or the Wiltshire study is how it's in the phenome database. In this study, we looked at 38 different inbred strains, and it was a one-day protocol. This was a battery of tests, but the open field was the first test we did on these mice. So they were naive when we tested them, and they were just placed in the open field, and we measured their behavior in the open field for 30 minutes. Some of the data I'll present will be for the first 10 minutes, where we look at kind of the anxiety, classic anxiety measures. The second study was done in my laboratory at the University of North Carolina. And this one had quite a different approach, because we were interested in looking at cocaine-induced locomotor activation. So these mice were tested over 3 days. They were naive, again, when we started testing them. On day 1, they were put into the open field after an injection of saline to habituate them to the environment and also to habituate them to handling and to the injection. Day 2, they received the same treatment, and on day 3, they got cocaine so that we can compare their response to cocaine. So, as I said, this was done in North Carolina, 45 inbred strains, most of which overlapped with the 38 we had done previously. We used standard open field equipment, at least standard for us, which is the Metassociates open field. This is the model number. It's actually the rat size. It's larger than the mouse size, so it's 17 by 17. The infrared beams are at 1-inch intervals. They're located in sound-attenuated cubicles with relatively dim lighting. It was, as I said, a 30-minute test in both cases, and we often look at the first 10 minutes to look at anxiety measures. So we measure distance in the open field, ambulatory episodes, rearing, also velocity or speed at which they travel, how much time they spend resting or not moving, and then also the location in the arena, whether they're spending time just along the edges or in the center. And so this just shows the results from both studies just as a strain distribution here. So along the bottom, this is the GNF study, the 38 strains along the bottom here. This is their locomotor distance in the open field. This is the UNC study, the 45 strains here, their distance in the open field. And I guess the one thing you can notice from this is that the data is kind of on the same scale. The range of phenotypic variability is similar in both studies. And this is distance. Distance was actually the primary measure that was first used when the open field was developed by Hall in 1934 as a measure of emotionality. Since then, many people have moved towards using this measure of center time, or how much time the animal spends in the center of the arena, which is considered to be an anxiolytic behavior. And this just shows the results from that as well. The percent time in the center, again, fairly similar across both studies. I'm not going to, it's hard to read kind of the order of the strains here, but I'll show that in just a second. And you can see the variability, and these are standard deviations. The variability in percent time in the center is quite a bit higher than the distance measures. So I want to say we made no real attempts to standardize at all across these two studies because they were kind of different. We did them for different reasons. So we weren't trying to replicate or repeat the first experiment. This was the second experiment was completely separate. There were some similarities. We had the same equipment, and actually it was the exact same equipment that I brought with me from California. The lighting's the same because they're in the same sound attenuating cubicles. We had used, on average, the same cage density, so the mice were group housed prior to testing. We looked at males in both studies, and I put shipping as a similarity because all of the mice were shipped. None of them were bred in-house, but I also include shipping as a difference because the mice we got in California were shipped from Maine all the way to California. The mice we get in North Carolina are shipped from Maine to North Carolina, which is quite a difference. The difference is obviously the geographic location and all the differences you have there, not the least of which being in California, there's quite a bit of seismic activity, which can affect animal behavior. Although in North Carolina, they started blasting for a building right next to our behavioral core when we started the test, so we had to avoid those days as well. The technicians were different. I was lucky, or maybe some people would argue unlucky enough, to have the same technician in each place do all of the animals, most of the animals for those studies. So within each study, it was usually the same person that did all of the testing. They were both female, in case there's a difference between male and female. The diet was different. The diet we use at UNC is different from the diet we used at GNF. The caging system was different. They were both ventilated cages, but they were different companies. And again, the major one was this experimental procedure. So for the first study, the mice were naive. They were just placed in the open field. For the second study, they actually got an injection of saline prior to being placed in the open field. So regardless of all of these differences and attempts attempts not to standardize, we see a really high correlation, which John talked about before, for locomotor activity across these two studies. This is the Wiltshire or GNF study and the Tarantino study. And you can see that for locomotor behavior, the correlation is 0.88. And for percent time in the center, it's 0.75. Probably not surprising because locomotor behavior and time in the center are often fairly highly correlated as well. But what we did notice, what I noticed, is that one strain in particular, C58, shown here, circled in red in both studies, always seemed to be highly variable within strain variants. And I started to wonder why that was the case, and I talked to Will about this because I knew he was interested in variants. I looked across all of the strains and looked at the correlation of variance within the strain. So how stable was that variation from study to study? And, you know, it was pretty high, but this was driven mostly by the C58 strain, which is always kind of the outlier is the outlier in both studies. When you take that strain away, we still saw a significant correlation of variance between studies, but it decreased quite a bit with removal of that strain. So now we'll go back to Will.
So when we're speaking about strain effects on variants, I think it's interesting to sort of think about the way that genes and genetic architecture might affect variants, and a concept that I was working with a couple of years ago was that of QTL for variants. And so this is an example of a quantitative trait locus for mean effects, and you can see, you change the genotype as a locus, and it changes the mean, and we might call that a sort of ordinary QTL, and we might contrast that with a sort of extreme different one, which would be a variance controlling QTL or a VQTL, as I like to call it, and one could imagine that there are genes that do sort of both of these things as well. And in previous work with a collaborator in Sweden, we sort of worked on methodology for identifying, for sort of jointly identifying QTL and variance QTL in common sort of genetic crosses, such as F2 crosses. But since we're on the subject of VQTLs and genetic effects on variants, I think it's interesting to say, how such increases in variance or decreases in variance might have come about, and use as a reference point Waddington's idea of canalization. So this was the idea that that the sort of genetic architecture of an organism evolves to create virtual canals, genetic buffering against environmental variation, and this is quite intuitive, I think, because everyone likes to think that they're special and different, and we're all a unique snowflake. But there are certain times in our lives when actually we really want to just follow the protocol, such as when we're sort of an embryo developing. We don't want variation. We want to really kind of buffer against it. And so there's an idea that sort of natural selection, or unnatural selection, such as inbreeding selection, favors this kind of stability. So, the sort of the genes are arranged so that within a particular environment we have this stability. And then if you move to a different environment, that canalized stabilizing architecture might not be valid anymore. It might actually not be stable anymore, and so you might get an increase in variance. And then you can also see something that looks like a genetically increased variance through something more like what John Crabb was talking about, where you have differences in mean effects with different environments. So through a collaboration between me and Lisa, we've sort of been thinking about, well, how can we explore variance genetics in this open field data? And one of the things we want to do is sort of prepare ourselves for using open field activity, for doing sort of gene mapping, gene prioritization by significance testing features across strains. But in order to do that, we want to be able to separate the contributions of batch and chamber and so on from those intrinsic to strain. And so tied in with this is the variances are hard to estimate in a precise way. I think it was John Tukey who said, he had a rule of thumb that you need 5 times as many observations to estimate a variance with the same precision as a mean. And so I want to sort of explore this using a model that would estimate these in a robust way. So I've been sort of playing around with a double linear mixed model, which is where so here we have a mouse I of strain S, batch B, and testing chamber C, and much like a sort of ordinary ANOVA, we have an effective mean, an effect of each of these things on the mean, but also we're simultaneously estimating effects of each of these things on the log of the variance, and we're employing shrinkage throughout through sort of hierarchical modeling, that these are random effects, not just for strain but also for batch and chamber, and there are some details which I won't go into, but one important detail is that we apply square root transformation at the moment to the activity data, and we're estimating this in the Bayesian way, so we can be quite flexible. When we do this, and wasn't really sure what was going to happen in terms of how similar the effects of strain and so on were going to be, but actually, so these are sort of 4 results from the 2 studies, but 2 different time points. So the UNC study looking at activity at 30 minutes, the UNC study looking at activity at 10 minutes, and then the same 2 with the GNF studies. And over here I have the sort of percentage variance explained of Y, which is the sort of common percentage variance explained. And then over here I have something which I don't really know what to call it, but it's a percentage variance explained of log sigma squared. So the percentage variance of the variance. And in both cases we see that there's a very strong effect of strain. And strangely enough, there's a bigger effect of chamber on the variance than there is on the mean in this case. Yep.
Before the transformation, after the
Oh, I see. Yeah. So these are just the sizes of those variance components. So yeah, there might be a little bit of kind of finessing if you were to transform back to the original scale. That's a good point. And so with a model like this, we estimate a lot of parameters, but because we're using a lot of shrinkage and so on, they're estimated in quite a stable way. And so these are the posterior means, and they're sort of Bayesian confidence intervals for the strain effect on the mean. And if we look at the effects on the variance, we see, okay, well, you're estimating variances, so you're not going to be as precise, but you can still see that there are strains that kind of move apart from each other in terms of variance. And then what in fact we didn't find very much of was, as I said, we didn't find very much effect of batch or of chamber. So if we look at this through a parallel lines plot, and this is maybe too small to see, but this is UNC at 30 minutes, UNC at 10 minutes, GNF at 10 minutes, GNF at 30 minutes. And what I'm doing is I'm connecting the strains to each other. And whenever you see a crossover, these are the strain means. Whenever you see a crossover, that's where they've sort of changed in rank. And you can see that actually the results replicate fairly well. There's a bit of change, but we also see that this is, so in blue, I should have written that, this is strain AJ, which is sort of the lowest mean strain, and then a relatively high mean strain is C58. When we look at the strain SDs, and remember this is not the same as the strain mean or the strain SD you'd get from just looking at the strains, because we're estimating through the model, so we're actually debatching, we're de-chambering, we're taking out all of those other effects. We see that actually AJ, even though it was quite low, has a relatively higher standard deviation, and C58 really just really is very, very highly variable strain, and Lisa's going to go into the sort of a deeper exploration of that.
So one of the advantages of using inbred strains or any reference population is the availability of data that already exists, both in the literature and in databases such as the mouse phenome database, which is housed at the Jackson Laboratory, and which Alyssa and her colleagues manage and build. And what I was able to do then, and this is, again, not a thorough search, but was able to go and search by C58 and see what other studies had used C58 in the open field and how those compared with our study, both in mean and in variance. And I found 3 studies. There was another Wiltshire study that was done with Matt Pletcher separately, and it was done in Matt Pletcher's laboratory at the Scripps Research Institute. There was a second study done by Matt Pletcher, again, with a different set of strains, overlapping strains, but an independent study, at least as far as the group of animals that were used. And then there was the Walston crab study, which John showed in his talk, that was done in, I think, 2004, at least published in 2004. And each of these strains used C58 in the open field. So what I did was first go and look at how these correlated across the different studies, and what you can see is similar to what John showed, right? So if you look at, this is the UNC study here on the y-axis, and the different studies here, the Walston crab. Pletcher and then the Wiltshire too. And you can see that there again is a very high correlation of locomotor behavior across these studies. But what I thought was striking is if you look and you're able to download the individual animal values from the phenome database, if you look at the variance of C58 in these studies as well, you can see that it always ranks as either one of the highest or the highest in variance in these different studies as well. So there's a lot of different reasons for variation or within strain variation, some of which may not be as interesting, at least to me, from a biological standpoint. But you can have age effects, right? So there are certain strains that have progressive type phenotypes, such as the AJ strain, which start having progressive locomotor phenotype. And if you're measuring across a wide age range, you can see quite a bit of difference in locomotor activity within that strain. You also have things like vision or hearing that can become progressively worse as animals age, which can affect their function in tests. Intracage aggression, specifically in males, differs between strains and differs between cages. You can also have what I call behavioral artifacts, and I don't think this video works, but this was provided, maybe it does, provided to me by Cheryl Moy, and this is actually the C58 strain. Maybe it doesn't. But what you can see in C58 mice, which are often used by people who are interested in autism, is that they have this stereotypic behavior, at least in the home cage, where they do a lot of backflips and what she calls jackhammers, which means they just jump up and straight up and down. And we would need video and observation in the open field to see if this may be the cause of the variability that we're seeing in this strain. You can also have, obviously, effects of previous handling, whether they're going through test batteries. In all of our studies, we do open field usually as our first test. I know that the Pletcher study, open field was done after a tail suspension test, so there was no other, or there was a previous handling in that study. You can have maternal effects. You can have batch effects that could be seasonal if you're testing across the same strain across a wide range of time. And then what we're interested in more is to kind of elucidate the genetic effects and find out if this strain really is more sensitive to the environment, which causes the variance that we observe. So I did go back and look in our data to see if in a non-locomotor-based test, did C58 also show this high variance or within strain variability. This is stress-induced hyperthermia, where the animals are given a rectal temperature, and then 10 minutes later, the same, and their temperature generally rises by at least 1 degree. And it's a measure of anxiety. And you can see C58 are definitely not at the high end of variance for that. This is corticosterone measurements, just basal corticosterone. And again, you can see that they're at the higher end, but definitely not one of the higher strains. So this implies that maybe it's specific to the open field, but we need to do a more rigorous kind of look across all of the studies that have been done with C58. I should mention that these were done in my laboratory, and they were part of a battery, and they were done later in the battery, so that could make a difference as well.
What if you're not really interested in looking for the genetics of variance? Variance and different strain variance is just a kind of a problem. And I was playing with the idea of, well, maybe we can describe different strains in terms of a common denominator, like a currency or something like that. So if we take say a very standard strain such as the B6 and we describe its precision, we might talk about the precision of some other strain as being some kind of equivalent precision animal count. That's just something I came up with on Friday. So we could define the EPAC of a particular strain in this way, and the way we might use that in experimental design is that for every, if we're looking at a number of strains, then if we know that some of those strains are just going to require more individuals in order to get the equivalent type of precision, then maybe we should budget for commensurately more of those strains. And when we analyze the data, maybe we should do kind of weighted analyses where if a B6 mouse counts as one observation, then a strain X mouse counts as EPAC observations, and just from the sort of modeling, I just give a quick chart of the EPAC, or the number of B6 I guess, that each strain is worth. And so you can see in the 30-minute GNF study, each C58 mouse is worth about 0.1 B6 mice. So it's just a sort of a thought. So in summary, strains differ in variance, and this can arise from multiple sources, including genetic background. The great thing about inbred strains is that you can have replication, so you do have ample opportunity for estimating strain variance, and there are a number of different ways that you might incorporate this into design and also into analysis, either sort of variance avoiding, or variance accommodating, or variance targeting. And so thanks to all these people.